So, what we've taught. Most of the bacteria will respire aerobically when oxygen is present, right? Most of the organisms that we deal with in this lab are either obligate aerobes, or actually the majority, as I mentioned earlier, are facultative anaerobes. We don't have any microaerophiles, but again, they would utilize oxygen, but remember at a very small concentration. Not easy to achieve in this lab, so we don't mess with those organisms. They will obtain a small amount of energy via glycolysis, but they will capture a relatively large amount of glucose's free energy from oxidative phosphorylation, right? Going that one step further, using the Krebs cycle in conjunction with the electron transport chain. And for these guys, when we say aerobic respiration, it's because they're actually using oxygen as that final electron acceptor. That's where the electrons go that they took from glucose. And so that's why it's called oxidative. It refers to the loss of electrons, right, by glucose that eventually go to oxygen. One thing at a time. They recycle their reduced coenzymes, those electron carriers, NADH and FADH2 that we talked about before. They pick up the electrons and then they carry them to the electron transport chain, and then the final acceptor is oxygen, which forms water. The carbon components of the glucose, that sugar, are given off in the form of carbon dioxide. Okay? So many bacteria can resort to fermentation when oxygen isn't present, right? So when there isn't oxygen, they're going to ferment the sugars instead. Remember, the major byproducts of fermentation are what? Acid, alcohols, and gases. Good. So this applies to our facultative anaerobes, right? When oxygen isn't present, they do fermentation. Your aerotolerant anaerobes actually always do fermentation, right? Obligate anaerobes can either do fermentation or they can do anaerobic respiration. And the difference here is at the end of their electron transport chain, instead of oxygen being the final electron acceptor, since of course they're living in an anaerobic environment, which means without oxygen, they're using other molecules to accept those electrons, right? Something other than oxygen. That's what anaerobic means. So they will capture a relatively small amount of the energy that's possible from glucose. Substrate level phosphorylation, making ATP with just the enzymes and not using the electron transport chain. So they recycle those coenzymes, as we said, by adding back those electrons to the products of glycolysis. In that process, they generate gases, alcohols, and acids. And there's lots of different possibilities, right, that we've looked at. By no means are we memorizing all those, right? We just need to know in general, when you ferment something, there's the possibility of acid production, always, sometimes alcohol, and sometimes also gas. So there's something in particular here, though, right? When we looked at our organisms, most of them can respire aerobically when oxygen is present, right? We've said that. Many can resort to fermentation when oxygen isn't present. Under certain circumstances, bacteria will prefer to ferment a carbohydrate, a sugar, even when oxygen is present. And we saw this in one of the tests, in some of the tests that we've performed. They've actually fermented the sugars, even though this lab is what? It's oxygenated, right? This is an aerobic environment. Why are they fermenting? There's oxygen, right? So that's a perplexing question, right? Why does a bacteria sometimes catabolize the carbohydrate source? Why does it do it aerobically, and other times why does it ferment, even when we have oxygen? So the answer is this interesting effect referred to as the Crabtree effect. So take Bacillus subtilis, for example. According to the chart, right, if you were to look at your chart of known data for this organism, it is an obligate aerobe, which means that it only utilizes oxygen. It's negative for anaerobic fermentation, right? It does not do anaerobic growth in the FTM. It's catalase positive, right? Again, confirming that this organism utilizes oxygen, lives in an oxygen environment, so it needs protection from those toxic forms of oxygen. So you have catalase, which breaks down what? Hydrogen peroxide into what? Water and gaseous oxygen. That's why we see that bubbling, right? So it means it has a functioning electron transport chain, again, does aerobic respiration. Why doesn't it use the electron transport chain and respire aerobically and gain more energy per glucose molecule instead of fermentation? We know that it does fermentation when you look at two of the tests. Phenol red test, right? A minus means that acid was produced. What color is that tube? It's yellow. So it fermented glucose. It can do aerobic respiration. Why is it fermenting the sugar? Right? And the Voges-Proskauer, the VP test, it's positive for that. What is that test for? It's another fermentation, right? But what's the product here? 2,3-butanol. What is that, an acid, alcohol, or a gas? Butanol. Ends in OL. It's an alcohol. It's an alcohol. So again, still a fermentation product, right? A particular type of fermentation. So why? Why is it fermenting this sugar? The difference is high concentration of sugar, right? So under these circumstances, right, they've got a lot of sugar. So they go the fermentation route instead of the aerobic respiration route. Again, why would you do that? You get more energy, right? So Peter has come up with an example. This is not a true mathematical example, right? But help, again, maybe make this make sense. So the difference is that it's much quicker to ferment a sugar than it is to send it through glycolysis, Krebs cycle, and electron transport chain. Granted, you get more energy, right? But it takes more time to generate that energy. Where, if you've got a lot of glucose or sugar, right, a carbohydrate, and you can go really fast through fermentation, even though it generates less energy, over a short time period, it would outcompete aerobic respiration. So here's a mathematical example to help us understand this. So suppose a bacteria can make 30 ATP for every glucose metabolized aerobically, but it takes 1/10 of a second to complete each cycle. So after an entire second, they've only generated 300 ATPs. And I say only because suppose now that the same bacteria can sense that there's an abundance, and they can, of glucose, they'll switch to fermentation. This only produces 2 ATPs for every round of glycolysis, right? Which is definitely less than 30, but it only takes them 0.005 seconds to complete a cycle. So they can do 20 times faster than aerobic respiration. So in a second, they can generate 100 more, because they can generate, in this example, 400 ATP per second, just doing glycolysis via fermentation. Right? So it is to their benefit, since there's a lot of sugar, just process it very quickly to generate a lot of energy very quickly, as opposed to going the long way where you're not going to generate as much energy. You're not as efficient, right? You're not getting all the possible energy when you ferment, but you are getting energy. And again, for these organisms, generating energy, right, the ones especially facultative anaerobes, being able to still survive in an anaerobic environment is better than dying, right? But again, depending on how much sugar is available, it's going to affect their growth, right? They'll grow slower if there isn't a lot of sugar. If there's a lot of sugar, they could actually even outcompete some of the aerobes because they can do it so fast. Does that make sense to you guys? All right, so that's why we see that effect. So how fast are enzymes in real life, right? This there's the study of how fast, right? How fast chemical reactions can happen utilizing certain enzymes is referred to as enzyme kinetics. So for certain enzymes, they've figured out, and you can see some of them are really fast, right? This one can produce products, 600,000 products per second. Others take a little bit more time. I mean, look at DNA polymerase 1, 15 nucleotides per second. That's still pretty fast, right? Lysozyme, which breaks up peptidoglycan, this guy's kind of slow. Can only really break, half a molecule in a second, right? So they take 2 seconds to break apart molecules, but eventually over time it can degrade it. Penicillinase, right? This is an enzyme that can destroy penicillin that some bacteria have, right? They can destroy 2,000 molecules of penicillin a second. So hence why, of course, if they have this type of protection, penicillin is not going to affect them, right? Because they're able to destroy it very quickly before it can affect their metabolism of peptidoglycan. So many enzymes are so efficient, right? So as you can see, very fast. If one substrate is consumed per reaction, one product is formed, right? So you take something and you produce something. In that case, LDH, which is a particular enzyme, converts pyruvate to lactate. This happens for us under anaerobic conditions, right? When we don't have enough oxygen, we do fermentation. We produce lactic acid, and that's why that can be a problem, because acids can be a problem for our metabolism. The turnover number equals the rate at which the substrate is consumed, which is equal to the rate at which the product is formed, right? So it's a balanced situation. Some of these reactions are very complex, right? And if this kind of stuff you're interested in and you think it's kind of cool, we have a new program at Delgado that I wanted to tell you guys about. It's the Science Laboratory Technology Program that's been approved. They're actually setting up probably the highest tech lab in this state, maybe even in this country. We're getting equipment in like mass spectrometers that community colleges don't have, right? This is something that universities have. It's a very growing field, biotechnology. There's a chemistry component as well. If chemistry is what really gets you jazzed up, but there's the biology component as well, which encompasses some things like biochemistry, right? So things like this, if these interest you, organic and biochemistry courses that we offer here at Delgado. And then the biotech program, that's what the BT, protein expression analysis, right? And actually looking at these proteins and how they work, most of the enzymes. That program is having an open house, and I'm going to post for you guys this week, and I'll post again to remind you. And you should have gotten a mass post through Canvas, but I know probably you guys don't read mass stuff that goes out. But the biology department is doing an open house. We're going to be utilizing my lab, that lab that they're setting up right now, in the A&P lab. But the focus is going to be on the biotech program. All the instructors that work in that program will be here that night as well as I'll be here. It's going to be, I don't want to say the wrong date. It should be the 19th, should be a Thursday in 2 weeks from 5 to 7. But as I said, I'm going to post the actual, there'll be light refreshments. If you want to talk to any other professors too, right? A lot of us are going to be here that night, so if you have questions about things, want to check out the new high-tech lab, come on by if you're on campus on Thursday nights. So that's a really cool program that we're excited to offer. And it's approved now, so it's an official program. So you can get an associate's degree. And highly sought after now for jobs in the biotech industry, because you'll leave here with all the skills necessary to go right into one of those lab positions. And there's an internship part of the program, so you might even work with a company and then get hired by the company later. And they've had some students that have already gone through the program that are already working in the industry. So really cool stuff. So today our lab focuses on the 3 plate media that we've kind of been looking at and using to help kind of confirm, your guys' results for your unknowns. So the Mannitol Salt Agar, I put a demo plate on each desk, right? So it's abbreviated MSA. And so you'll see there's 2 sections of growth, and then 1 section I didn't inoculate. So you can see that, again, the media started out this color. Then I also have an EMB, right? This one, you're going to actually want to open it up and look at the actual growth on the inside. That's why I have it hinged and wrote the initials on the top. The blood agar plates I kept up front because I got a whole bunch of stuff on one plate, and so I want to explain it to you guys when I pass them around. And yesterday I got distracted and I forgot to do one of the inoculations on one of my plates. So, oh well. But I have examples of everything to show you guys, too, for each of these media. So, all 3 of these plates are what we refer to as selective media. They all select for the growth of a particular group or type of organisms based on components that in the media that usually inhibit the ones we don't want growing, right? So there's all of these have some type of selective agent, right? They have something that's inhibiting the growth of the organisms we don't want, therefore selecting and allowing the growth of the ones we are interested in looking at. And then these media go one step further in that they're differential. So if in fact they grow, we know an additional characteristic, a biochemical ability. And for the most part, right, we've been dealing with fermentation in this class. So for 2 of those plates, again, we're looking at sugar fermentation. The other plate, the blood agar plate, we're actually looking to see if they can destroy the red blood cells and to what degree. So when we're selecting, obviously, if your organism didn't grow, we can't ask the differential question, right? Only if it grows, then we could differentiate for the second question. So all of these plates, you have 2 results, right? You have the selective result, and then if it grew, you have the differential result. So MSA is our first one, and that's a selective media. And what group of organisms are we selecting for? Typically, we're looking for Staphylococci, but the selective agent in this media, do you guys know what's toxic to the other organisms? Nope. Yeah. It is the high salt concentration. This is important because most media has salt in it for isotonic balance. This media has a higher than normal salt concentration. So although mostly we look at Staphylococci, it does not mean, as some of you guys saw, you had growth, right? So Amy had growth on MSA, but she doesn't have a Staphylococci. But since it grew, what do we know about your organism? It's salt tolerant, right? So in clinical specimens, typically they're looking at Staphylococci. But we don't have just clinical specimens in this lab. You have a soil microbe, right? Which apparently is also salt tolerant. Okay, so, and I'm not kidding when I say high. It's 7.5% salt, where saline, which is isotonically balanced, is 0.9%. Right, so this is very high, which is reminiscent of what environment for our bodies? What's highly salty? Sometimes. When you sweat. So therefore what becomes salty? Your skin. Right? Your skin can be salty. So it's also differential. So if you have growth, then we can ask the next question. And what carbon compound in the MSA makes it differential? What's going to get changed by these organisms? So it's called Mannitol Salt Agar. We already talked about the salt, so what are we left with? Mannitol, right? Mannitol is a sugar alcohol. This is a sugar in its alcohol form. So, as I mentioned, some organisms can ferment this. So again, they're going to convert it into something that can go into glycolysis, generate pyruvate, but to regenerate those electron carriers, they're going to produce acids, alcohols, or gases, which is the easiest to detect in media. The production of acid, alcohol, or gases. Acids, because what can we have in media that's going to help us detect acids? What can we add to media to help us detect acids? A pH indicator. What's the pH indicator in these plates? Phenol red. You guys dealt with phenol red before, right? So what is acid production? What color is it going to be? It's going to be yellow. So this is a beautiful streak plate that was done several semesters ago by one of my students. So you can see definitely where there's a lot of growth, it produced a lot of acid, lots of yellow. But here, the plate is kind of orangey because there's not as much growth, so it hadn't produced a lot of acids to completely turn it yellow. I just did spots on the plates, and you guys did too, right? We didn't do streak plates. But you can see, as I said, right, that entire section turned yellow where it fermented mannitol. So you guys all have, right, this plate, you all have a yellow section. So we know that organism, it grew, is salt tolerant. And we also know because of that yellow color, it fermented that mannitol, that sugar alcohol, and produced acids evident by the yellow color change of the phenol red. So what is the name of the organism most commonly found in clinical specimens that can grow on this media and in the process generate the acid byproducts? Staphylococcus aureus. Right? And so that helped confirm Eden's results. She had Staph aureus. What purpose does Nutrient Agar plate serve? So we didn't do this. In what ways does it increase the validity of our test? So, what is Nutrient Agar? It's just plain old growth media, right? All your organisms or TSA grow on it. And they don't look different or anything, per se. But if we streak them onto MSA and Nutrient Agar, it kind of acts as a control, especially if it didn't grow on MSA, right? You know it really just doesn't grow because of that high salt. It isn't because you had a bad inoculum if it still grew on your Nutrient Agar. We didn't bother with this, right? And they wouldn't do this in a clinical lab either. And the reason why we didn't bother with this is because you guys have been all along propagating your organisms on Nutrient Agar and TSA. What would happen if we omitted the salt? This media would no longer be what? It would no longer be selective, right, for salt-tolerant organisms. So therefore, if the salt isn't in there, just about any organism would grow, right? And you would be confused and you would end up with false results because you would think maybe it's a Staphylococcus or salt-tolerant organism. So that salt is very important to the selective ability of these plates, that high salt concentration, remember. It's not just the fact that salt is there, it's the fact that it's a very high salt concentration. Right? So you would end up with false positive results. You definitely don't want that. So will this affect specificity or sensitivity of this type of test? Specificity. Again, it will no longer be specific for salt-tolerant organisms if you remove the salt. So all organisms need a carbon and nitrogen source. This was very evident in the citrate test that we did, right? And that we only gave them citrate as a carbon source, and we gave them only one nitrogen source. All organisms need to have both in order to grow. So in this media, there are there's plenty of carbon and nitrogen in the beef extract. Because all beef extract is cow cells, right? And so they're animal cells. They contain all the nutrients that should be necessary for carbon and nitrogen sources. In addition, peptone, which is a protein source, which contains both carbon and nitrogen, is in the media. So there's plenty of things for them to eat. Mannitol is used mainly as an energy source, if they can utilize it. But it only contains carbon. So they can produce energy from carbohydrates, from the mannitol, if they have the necessary enzymes to break down that particular sugar. And the reason why we usually catabolize sugars is they actually generate more energy than when you break down proteins. Okay? And especially with proteins for our bodies, you can produce acidic products from the breakdown of protein, so you don't really want to do that. But if you absolutely have to break it down for energy, we can. So with the diversity of microorganisms in the world, how can a single test such as Mannitol Salt Agar be used to confidently identify Staphylococcus aureus? Can't, can it? Right? It could help you narrow it down for sure, and especially if you're dealing with clinical specimens, if you got this type of result, it probably is Staph aureus. This would be a presumptive, right? We were at this point. But again, depending on the need, additional testing would be done, right? So some of you guys, like Eden, is probably like, man, could I just started with mannitol salt and then, bam, I would have known that was my organism, right? But not necessarily, because, you know, other organisms could potentially produce that result. Just in the group that we're looking at, this is pretty much the only one. So what can you tell me about this plate? Do you see growth? Yes, right? So what can you tell me about this organism since it's growing on Mannitol Salt Agar? It's salt tolerant, right? Otherwise it wouldn't grow. It would be killed by the salt. Did the media turn yellow? No. So is it fermenting the mannitol? No. So this one's not fermenting the mannitol. So it could be a staph, right? Because it is salt tolerant. And in fact it is, because I know what was put on this plate. It was Staph epididymis, which makes sense. It's named after the fact that it can survive in the dermis of our skin. Again, it has to be salt tolerant in order to do that. But this one tends not to be pathogenic, right? And one of the ways to distinguish that from aureus is the fact that this one does not ferment mannitol. So you'll see on those plates, the one that didn't change the color of the media is Staph epi. As we said, if it was Staph aureus, though, it would have fermented the mannitol. You would have had a yellow plate. So here's the two of them side by side, similar to the demo plates I have for you guys today. So both of them grow. Both are salt tolerant. Both are Staph, right? One can only ferment the mannitol, and that's Staph aureus. And that's the one we worry about with pathogenic infections, because a lot of Staph aureus can produce really damaging toxins and enzymes that can be very destructive. And we've got problems with antibiotic resistant strains as well. So, although this is not a great picture, right, some of our organisms, like Amy's, will grow. They are salt tolerant. Sometimes over time they'll even start to ferment the mannitol. So notice that this side of the plate is kind of orange because that pH indicator goes from pink, right, to kind of a reddish color. Orange is neutral, and then yellow is acidic. So the fact that this one is going from pink to orange, it's producing a little bit of acid, so it's fermenting a little bit of the mannitol. So, scientific method, right? Why are we doing this? We want to know if our organisms are salt tolerant, what we call halophilic, or haloduric, which is salt tolerant, where halophilic are actually salt requiring. Our Staphylococci are salt tolerant. Some of the bacilli And again, bacilli wouldn't be in a clinical specimen from a person. Among the survivors of the selective media, then we can ask the question, can they ferment the mannitol? Right? If it doesn't survive, we can't even ask that question. So we just put them on the plates at optimum temperature for 1 or 2 days, and then I refrigerated them to stop growth, so we wouldn't get false readings. So media surrounding the colonies remains red. That means no fermentation happened, right? Media surrounding the colonies yellow means that fermentation happened. And only if you have growth does it tell you something, right? It tells you whether they're salt tolerant or not, and then from there we can know if they're fermenting. And this is really indicative of Staph aureus. So then we get to the CNA, and sometimes in some of the handouts I give out, the stupid autocorrect makes this CAN, but this is important. The abbreviation is CNA, and it's for the 2 antibiotics that are in this media. This is the particular formula of the media. It's Columbia, and it has 5% whole red blood cells in it from sheep. Sheep red blood cells are very similar to human red blood cells. It's one of the main reasons why they use those. And also not to deplete the human red cell supply, right? So we use a different animal. So these plates, we're really looking for if the organisms can produce hemolysis. And as the name implies, these are toxins released by some bacteria, and they have the ability to lyse red blood cells. So hemo referring to blood, lysin referring to the destruction of those cells. So why do some organisms produce hemolysis? So they can do what? Lyse red blood cells. Okay, so what? What element inside that red blood cell are they after? They're after the iron. They're after the iron. They need it just as much for their metabolism as we need it. It is the major component of the hemoglobin protein inside our red blood cells that helps us carry oxygen. But microorganisms need iron too for their metabolism. So our red blood cells are a very rich source. So if you can get at that source, right, if you can lyse red blood cells and get at that source, that's beneficial, right? So this is a selective media for what group of organisms? Gram-positive. And seriously, it was like, even if you didn't do your homework, you had a 50-50 chance of getting the bonus right today, right? So gram-positive, and the reason for that is there are selective components that stop gram-negative. The selective components are colistin and nalidixic acid. Don't worry, I won't make you spell them, but you do need to recognize them. Okay, so that's what CNA stands for. C for the colistin and Na for nalidixic acid. These are 2 antibiotics that affect gram-negative. So E. coli is a gram-negative. So notice, too, the similarity. Coli, colistin. So this media is also differential. What component in the media gets changed by the bacteria? The red blood cells, right? And they are whole red blood cells, which is why those plates are blood red. So if it grows, we know it's gram-positive, right? Because gram-negatives are going to be killed by those antibiotics in these plates. And typically we're concerned with gram-positives that have this ability. Not to say that some gram-negatives don't have this ability. Some of them do. But mostly, especially in clinical specimens, again, we're looking for the gram-positives, which typically are going to be quite pathogenic. So there's 2 types of hemolysis that can happen. What we refer to as beta, and that's where it completely destroys the red blood cells. So for these plates, when you hold them up to the light, you can see complete destruction of the red blood cells near the growth of the bacteria. So I have a couple of excellent examples of that I'm going to hand around in a moment. And actually, Eden's plate has a really good example too. So pass this around, hold it up to the light, and you can see that clearing, that complete clearing around her plate. Let me pass that around. Alpha, on the other hand, implies that it's not complete. It basically pokes holes in the membrane. When that happens, oxygen gets in, and that oxygen reacting with the iron creates this greenish discolorization, almost like when iron rusts, right? How I remember the two is I always remember B for beta, and I think bad, right? This is the worst case scenario for what's happening in our bodies for our red blood cells to be completely obliterated. Not to say that alpha isn't bad too, but it's just one of those play on words that can help us remember. So, for these two plates, these are my older ones, there's a really cool effect that's happening too. And again, hold these up to the light. You'll see that one of these has got a label on the back that says BC. That's Bacillus cereus. And this is a spore former. So now that the plate is aging, the cells are starting to form spores. So you don't have as much growth getting in the way, and you can really see that clearing. Now, Staph aureus, on the other hand, is beta hemolytic as well, but it's kind of harder to see sometimes. It isn't as aggressive as the bacillus, and you can see its growth is still maintaining right now. And then there's a cool effect where I think some of the lysins are diffusing from both of them, and so there's a really cool zone of very hemolytic. And again, you can see right through the plate where it's lysed. And then you'll see one that's labeled M. This was actually a swab I took from my mouth. A lot of us have streptococci mutans associated in our mouth. You just can't avoid it. It's part of our normal flora, but unfortunately it's the one that contributes to cavities because it ferments the sugars that you eat and produces acids which deteriorate the enamel of your teeth. So these organisms are the alpha hemolytic, again the greenish, and the reason why I use a mouth culture is because we would have to use blood agar plates to maintain most of the alpha hemolytic bacteria out there. We would have to keep growing them on this blood agar plates. These are kind of expensive. When I worked at a different school, not to be named, it was associated with the hospital, and so I would get a sample from the hospital all the time of Streptococcus pneumoniae that is really good alpha hemolytic. But one of our smart colleagues here was like, y'all, we have it in our mouths, right? We can just use our mouths to culture and our students can see alpha. So there's alpha, and you can see it's really green. And then the other ones that are on the plate, I had put EF, Enterococcus faecalis, because it's supposed to be alpha. I have to say it doesn't look very green to me, but who knows. And Staph epi, which of course is gram-positive, so it will grow on these plates, is not hemolytic. So you'll notice there's no lysing for that one, and that one's a radiated SE. Now I'm noticing some of my tags are coming off, so I'll rewrite them so you guys know what's what. This is SA. For Staph aureus, this is Staph epi. So share this with that group. I'm going to give this to you guys. This one got that really cool effect. Take a look at all of those and then pass it on. I guess I'll swab my mouth and stick this one back in the incubator. Can't believe I forgot to do that yesterday. Okay. Cool stuff, huh? Okay. So again, here's some more alpha. Notice the greenish tinge to these plates. So for this one, it's growing, right? So it's gram-positive or gram-negative? Gram-positive, because remember the antibiotics would kill gram-negatives. What type of hemolysis are you seeing here? This is beta. And then if you can read too, notice this was a Staph aureus plate. Right. And again, this is one where they did the streak plate and came out awesome. And you can see right around the colonies that complete clearing. So beta hemolysis, complete clearing, and of course it's gram-positive because gram-negatives would be killed by the antibiotics. Then you have a third possibility. It can grow, but not actually lyse the red blood cells. So if it's growing, we know it's a gram-positive, right? Which is the case for Staph epi, right? That's a gram-positive, but it does not lyse the red blood cells. This is referred to as gamma, which I hate. Gamma lysis, it sounds like it's lysing the red blood cells, right? But gamma actually means no lysis, right? So you'll see growth, but you'll see no change in the red blood cells. And so my example of that, as I said, you see that Staph aureus on there. It's growing, but it doesn't affect. There's other things in that media for a food source. So they can grow. Just some of them really require or grow better with the iron that they can get from the red blood cells if they produce those hemolysins. So this was an interesting plate. The student has a gamma organism. It's gram-positive. It can grow on this plate. Or actually, this semester we didn't have any CNA. You'll notice that there's no CNA here. So this was just a blood agar plate. It didn't contain any antibiotics. But you can see there was definitely some contamination here in her plate, right? This abnormality here isn't what you see on most of the plate. This is in her organism. This is a contaminant that happens to be what type of hemolysis? Beta. And this is Staph aureus, right? And I would guess it came from her nose, from breathing on her plate, right? Probably didn't cover up her plate with her lid as she was doing this, right? And so sometimes even when I'm inoculating plates, I'll hold my breath for a second because a lot of us carry Staph aureus in our nose. It doesn't create a problem for us on our nose. It's when it infects our skin and stuff like that that can cause some serious problems. She literally breathed on it. So like air from her nose probably got onto the plate that contained this organism. Yep. It's a very common contamination that can happen with these plates. Which is, again, why I said typically when I spot inoculate, I hold my breath for a second. Because I don't want to contaminate it with Staph aureus if I have that in my nose. Okay, most of us do at one time or another, and it's not pathogenic for you in your nose. And you may not even have a pathogenic strain. It might not be producing those other toxic enzymes and stuff like that. But sometimes it could be. And that's why, like, staph infections and stuff, they usually, if you have a serious wound, right, it could come from your own normal flora, right? It could have come from you. These are highly prevalent in the environment. A lot of us have it. So this is a plate again. You can see right through it. So what type of hemolysis? This one's beta. And again, what do you see here? You can see right through it. That's beta. This, no change to the red blood cells, but you see growth, right? So that's gamma. Here's the greenish, so that's alpha, right? And again, this is beta, just not as strong as this one, but you can see these zones of clearing that's happening. Here's the two staphs side by side, right? You can see the staph again, beta hemolytic, right? And staph epi not. And again, you see growth, but no change to the red blood cells, right? So this is an example of gamma, which is no hemolysis. So Many, but not all, species of Streptococcus are beta hemolytic. An additional test that they'll sometimes do, you'll see these marks that actually poked into the agar, so that it created an anaerobic type environment. Some of these guys, under anaerobic conditions, these Staphylococci, produce more hemolysis, or they're even more effective under anaerobic conditions. Additional tests that they'll run are tests that will test for the different polysaccharides of sugars associated with the cell walls of Streptococci. This testing is referred to as Landsfield classification. And this is similar to the type of testing that we're going to do the very last lab of this course, where we're going to use antibodies to detect the presence of antigens. And that's what they do for the Streptococci. And even the hemolysis they produce, they give them a special name. Sometimes they refer to as streptolysis because they're produced by Streptococci. So as I said, some of them won't work in oxygen. Some of them will work, but then work even better under anaerobic conditions. So that's why they would poke it into the agar. So you can see that lysis, right, where they poked it in. So alpha under aerobic conditions, see the greenish? But anaerobic, where they poked it into the agar, see how it's clear? That's beta hemolysis. So many but not all Streptococci are beta hemolytic, right, where they completely lyse the red blood cells. So serological classification, again, is utilizing antibodies to detect particular sugar antigens on the surface of these. So there's lots of different groups. When they refer to the groups, right, group A, group B, that's Landsfield testing. And so give you some examples, and this guy is really strong beta hemolytic. Streptococcus pyogenes is actually the one that causes strep throat. So when I was a kid in doctor's offices, they literally would swab your throat and rub it right on a blood agar plate. Doctors don't do this kind of testing in their offices anymore. They swab your throat, stick it in a tube, and send it off to the lab. And somebody in the lab puts it on a blood agar plate. In addition, now we have the ability to test again for the antigens using antibodies and little quick assays. It's called the rapid strep test. So they can test for the presence of the antigens, the bacteria, from a swab sample in a little quick kit in the office. And this utilizes antibodies. You can sometimes get false negative results, which means that it comes out negative but you still have the infection. So they're always going to send the swab off to the lab for confirmatory. But the good news with that is that typically they called you the next day and told you your results of your culture, and if it was positive you had to take your antibiotics. Now, if they can get a positive result within seconds, they send you right off to the pharmacy and you pick up your antibiotics and start treatment sooner. Our immune system can eventually usually take care of this type of infection, but anyone who's had strep throat, you don't want to suffer through. And this one can cause pretty serious side effects depending on the Streptococci strain of pyogenes you're infected with. So we usually don't want people to not take medication when it comes to this type of infection. So other ones that we worry about, sometimes they refer to as the beta strep test, are ones that women, we have some Streptococci that will infect our vagina, and again, can be part of our normal flora. It doesn't cause any problems for us in the vagina. The problem is that when you give birth, this could infect the baby. And so they'll test you right before they'll culture swab from your vagina right before you're getting ready to give birth to see if you're positive for this normal flora that could be damaging to the baby, the Streptococci. And they'll give you antibiotics to prevent passing it to your child during childbirth. So, again, it's the sugars associated with the cell wall, and these guys are all encapsulated as well. So that adds an extra level of difficulty when it comes to our body being able to identify. So again, a lot of them are beta and alpha hemolytic, and the Landsfield group is a different classification. It's based on these sugars. So notice, as I said, Streptococcus pneumoniae is alpha hemolytic, but it's really dependent on those red blood cells. And pretty pathogenic too, so it's another one of the reasons why we don't like to use it in the lab. And in fact, the company that we buy our organisms from to utilize in the lab, they will only sell these pathogenic ones to universities and colleges. They won't supply them to like high schools and stuff like that may be doing microbiology because of the added risk, right? So again, we don't do the Nutrient Agar, but it would provide extra data. It would show that your organism can still grow if maybe it didn't grow because of the antibiotics. But we didn't do this. So early formulations of this had a slightly higher concentration of the two antibiotics, colistin and nalidixic acid. So colistin is an antibiotic that affects membrane integrity. And remember, gram negatives, how many membranes do they have? Gram negative, how many membranes do they have? They have two. They have that outer membrane, right? And so they're very sensitive to colistin. Think of E. coli, gram negative. Very sensitive to this because they have that outer membrane that's disrupted by this antibiotic. And then the other antibiotic, nalidixic acid, affects DNA replication, but the enzymes that are used for DNA replication are different in gram positives versus gram negatives. And so the target in this case happens to be one that gram negatives have and gram positives typically do not. But if you use too high of a concentration of anything, it can be toxic. So I always use the example of water. You can die of water toxicity. If you keep drinking water and you never pee, you can actually die from too much water. So anything in excess can be toxic, right? So if the levels of antibiotics are too high, it could even kill off your gram positives. So it's important that the concentration be a good level, that we're just killing off the gram negatives and we're not affecting our gram positives. Because remember, we want to select for the gram positives with this media. So as I said, remember, gram negatives have that additional outer membrane and are really sensitive to the colistin. So our scientific method, right? Why are we doing this? We want to be able to grow our gram positives, which helps confirm for you guys your gram positive result from your gram staining. If they are gram positives, we want to be able to differentiate whether they can do beta, alpha, or no hemolysis at all, referred to as gamma. We want to be able to differentiate. So the last plates, which are my favorite, because I like the pretty colors that they generate. So this media is selected for what group of organisms? This, we're looking for gram negatives. What components in the media are therefore toxic to our gram positives? EMB stands for eosin methylene blue. Those are two dyes, right? We've used methylene blue, right? Eosin is a red dye. So that's why these plates are kind of purplish, is because the dye is present. And these dyes are toxic to the gram positives. They don't have that outer membrane to help protect them. So these dyes get in and they're toxic and they kill the gram positives. So again, if you had a gram positive, it shouldn't have grown on these plates. Only if you had a gram negative, this should confirm, right, and would have grown on these plates. Then, once they grow, these are differential plates as well. They help us differentiate an ability. What can be fermented in these plates by organisms that grow on it? Sugar, right? Gets fermented. What sugar's in this media? According to your lab manual, it could be lactose and sucrose. But the formula that we utilize in the microbiology lab here actually only contains lactose. So we're only asking the question, can it ferment lactose? Sucrose is the same thing as table sugar, which is fructose and glucose hooked together. It's a disaccharide. But we're just looking at lactose. So why do we use this type of media? Typically, this type of media is used to look for coliforms or fecal coliforms. So coliforms are a broad class of bacteria found in the environment, including in feces of humans and other warm-blooded mammals. So coliforms are lactose fermenters. So they're going to grow on these plates and they're going to ferment the lactose. They're, again, gram-negative, so they'll grow on these plates. They tend to be bacilli, rod-shaped. They also produce gas as well as acids from lactose fermentation. But again, we're not going to be able to detect the gas in this case, just acid production. Fecal coliforms, right, ones that come from feces, are lactose-fermenting gram-negative rods that reside specifically, right, in the intestines. So therefore, they're commonly found in things that have been contaminated with feces, with excrement. So they're defined as enterics, right, enteric meaning from the intestines, that produce acid from gas, acid and gas from glucose fermentation. At around 44 degrees Celsius, plus or minus 0.2 degrees, and after about 24 to 48 hours, around 24 hours. This is a relatively high temperature and responds to 112 degrees Fahrenheit. We don't necessarily do that higher temperature. Normal 37 body temperature is usually sufficient. The presence of coliforms, right, in our drinking water may indicate a possible presence of harmful disease-causing organisms, right? Because we know feces can contain ones that could make us very sick. So approximately 90% of fecal coliforms, especially E. coli, and other genera are present, which include Klebsiella, right, and Klebsiella pneumoniae that we've worked with, right, be pathogenic, and Citrobacter is another genus that can be present and are pathogenic in feces. So when the pressure, right, of New Orleans water purification plants drops below 15 psi, they issue a precautionary boil water because our drinking supply may have been contaminated with sewage, right? Therefore, fecal coliforms and could potentially make us sick. That's why they're telling you to boil your water. What you're waiting for, why we have to wait for them to tell us that, oh, okay, it didn't actually get contaminated, it's all good, is that they're going to take water samples, several water samples. They're going to run them through a filter, a sterile filter system that'll trap the bacteria. They're going to put them on plates very similar to the EMB or McConkey agar, which is another media that, again, is going to promote the growth of gram-negative lactose fermenters. There's a pH indicator that helps us differentiate. And so the allowable amount in potable drinking water is 1 coliform per 100 milliliters of a sample, right? So it's okay if like 1 happens to be in there. This is not 1, right? This type of result would have been, okay, water really isn't good, keep boiling and we'll fix the problem, right? The good news is that over the past several years, the 5 different times we've been told to do this, all the samples have come back negative, right? And even if you didn't boil it, you would have been okay. But you want to boil it just to be sure, or drink, bottled water. So where can you find coliforms? They can be in the soil or water, right? And it really is the ones that come from feces that we're concerned with, that can be pathogenic, right? The ones that are enteric, that come from the intestines. E. coli isn't really usually very pathogenic, and all of us have got it. It's when that strain has different toxins that it can produce that can make you really sick. But as we know, Salmonella can make you very sick, right? Shigella can make you very sick. Some of these other genuses contain really bad bacteria. Notice some of these are gram-positives as well that can cause problems for us under certain circumstances. So no, coliforms are found in the environment, right? Both places, as well as in the gut. But fecal coliforms are coliforms that are only found in our gut, right? So really are a strong indicator that that type of contamination has happened either to a water source or a potential food source. So, and E. coli is a really good indicator because it's only found in our intestines, where some of them, right, can be found in both places. So, Klebsiella can be in the intestines, but also can be in the environment. And same thing with Citrobacter. So how are these guys related? Again, some of them are non-pathogenic. Others are opportunistic pathogens, right? Especially E. coli, Klebsiella proteus. When these guys get in other parts of our body, out of our intestines, they can cause some serious problems. So there's one strain of E. coli known as E. coli 0157 and has developed the Shiga toxin. It probably got it from Shigella, which has this very potent toxin that can be very destructive to the intestines. But not all E. coli has that. So, sorry about this picture. I didn't pick this one. Peter did. Unsanitary slaughter practices are one of the ways that our food can get contaminated, right, with fecal coliforms from the animal that's being slaughtered itself. Water and soil contamination, right, so can contaminate our crops. Direct contamination by food handlers, right, or even ourselves or preparers. How do we contaminate our food? By handling it, not having good sanitary practices. This is why every bathroom you go into in a restaurant, it tells you the employees must wash their hands before they leave, right? Staph aureus, which is actually a gram-positive, some strains can produce an enterotoxin, entero again referring to our gut, and cause some really serious problems. So not a coliform, right? And causes a particular food poisoning known as topidine. And this is some background on that particular substance. So my favorite story is good old Typhoid Mary, whose name was actually Mary Malone. She's called Typhoid Mary because she was a carrier of Salmonella typhi. So she had this infection. And what actually happens with the carrier state is that this infection can get into the gallbladder and grow there. And what happens is that as the bile is excreted into your intestines to help with the digestion of fats, it's also secreted in this bacteria. So this bacteria was getting into her feces. And apparently she wasn't a very sanitary person because she worked as a cook, and several people got sick by the salmonella because she contaminated the food she was preparing for them. And so they linked it back to her and they quarantined her. And back then they basically jailed her. Okay? After several years they released her. They told her not to work as a cook. What did she do? She went and worked as a cook in a maternity ward. So again, a whole bunch of people got sick. They linked it back to her. They quarantined her again, basically imprisoned her until she died. What could we have done to rectify this problem? Well, back then we didn't have antibiotics to clear up the infection. And quite honestly, with this one, I don't think it affects, like, if you have the carrier state, I think they remove your gallbladder to get rid of the infection. It's the only way that you can actually get rid of it. That just stores excess bile. You still produce bile all the time by your liver. You just can't eat really fatty meals because you can't pump that extra bile from your gallbladder into your intestines really quickly, right? You have to produce it. So, what could we have done for this poor woman? They say, don't be a cook. Well, she's an immigrant from what? Ireland. What does she know how to do to make a living? Cook. So what do we need to do? Teach her how to do something else, right? Also, what could we teach her to do so she could still be a cook? Wash her hands after she goes to the bathroom. Sanitation, education, right? Very important, right, to the ability, right? So later on, right, they found she had the carrier stage. She didn't know she was sick, right? But she also didn't know good sanitary practices. We know better nowadays, right? Like I said, every restaurant you go into, there's a reason for that, right? You don't want them contaminating your food. So Salmonella and Shigella, right? There are specialized media tests, again, and even for E. coli, that help us determine if these things have been found in different samples. Bile salts, think about this. Alrighty, we'll look at all that later. Are detrimental to some microorganisms, but of course the ones that live in our gut have to not be affected by that. So bile salts can even be toxic to a lot of the gram positives, because remember, a lot of the enterics are gram negatives. So that's in a lot of the media, and again, a lot of times it's going to have lactose, some type of pH indicator, so we can determine if it's fermenting that lactose. Some of them have iron added, because as we said, iron is necessary. In the case of this one, that's why it turns black, because of the utilization of the iron. So E. coli is a fecal coliform. What color would you expect it to grow on the plates? It's going to be that black and to metallic green. So you have a little bit of metallic green, right, on those plates. It's a dead giveaway that that's E. coli. Of course, that's not the only one, right, that can do that. And notice again, we're just seeing that it's fermenting the lactose and producing the acids. The acids interact with the dyes in the media, and that's why you get this color change. So if an organism ferments lactose but produces a small amount of acid, E. coli produces a ton of acid, which is why it's that particular dark color and pretty color that I like. Others will have a pink or purplish growth. So notice the colors here. If it doesn't have a distinctive pink or purple color, then it's said to be negative for lactose fermentation. We have a couple in this lab that are supposed to be negative, but it's really hard to tell. I actually got a pretty negative result for one of these. So on the side that's labeled PA, you can see there's really no colorization, but it is growing. It is a gram negative. This is Pseudomonas aeruginosa. And this one is supposed to be negative for lactose fermentation. And this really looks negative. It's probably the best negative result I've gotten. So again, Nutrient Agar would just be a control. Wouldn't do it in a clinical lab setting. We didn't do it because we've been growing on that media all along. So dipotassium phosphate is a buffer that's added to EMB, which helps adjust the pH. Because pH is important, right? Because what do acids do? They lower the pH, right? And we're measuring pH by these color changes in the interactions with the dyes. So if the pH is higher than it's supposed to be, that organism is going to have to produce a lot of acid for us to get the different color changes. So we could end up with some false results, right? We won't see maybe that black or metallic green of our E. coli because the acid-base balance wasn't at the right beginning starting level. It was too high. So although 0.8 doesn't seem like a lot, that is it can significantly affect the reading of these results if your pH isn't adjusted correctly. So unlike the mannitol salt, which use a pH indicator, so we can kind of tell when we first look at our plates that it's at the right pH because it's a pinkish color. With the EMB plates, you can't tell by just looking at the plate that it's at the right pH because it's not necessarily a direct pH indicator. We're using dyes, which then also interact with the acids, but this is also why for these plates, you've got to look at the organism's color of their growth, not the plate, unlike MSA where you're actually looking at the color of the media.
So if it's pink or purple, it would be a moderate?
Yeah. So what would be possible consequence, right, of having too high of a pH? We could end up with false negatives, right, which this affects the sensitivity of the test, right, because we can't really tell what level of fermentation is happening because the pH isn't adjusted right for the starting value. So again, if it's metallic green, chances are, right, if you're in Antarctica, it looks like a penguin, it probably is a penguin, right? It probably is E. coli, but again, remember, not just one test. You would use additional testing to confirm if you really need to know what the organism was. But pretty strong indication. So a couple more pictures. Are these organisms gram-positive or gram-negative? Well, they're growing on eosin methylene blue, so they are gram-negative because the dyes kill off the gram-positive. Are these lactose fermenters? This one, you don't see the pink or purple. It's actually kind of blending in with the plate like the one I sent around for you guys. So this one would be non-lactose. This one is pink, so it would be a little bit of lactose fermentation. This one is pretty much the same color of the media as well, so that would be no lactose fermentation. So notice kind of pink, kind of dryish. One of our organisms was growing kind of like this before, but it didn't this semester. I don't know why. But again, notice the pinks here. Right? These are all gram-negatives because they're all growing. Right? Pink indicating lactose fermentation, and notice this one's got the metallic green. So that's pretty unique, probably E. coli. And then our scientific method. Alright, so if it's growing, it's gram-negative. If we get color changes, depending on what the color is, pink would be modest, right? Pink to like this purplish color, right? And definitely black to metallic green, very strong fermenter. And for this group, it's just E. coli that does that.